All right, good morning, good afternoon. Welcome to our third installment of the Biological Magnetic Resonance seminars. So today we have two dynamic iono system professors telling us about their work in applying solid state NMR and solution NMR to emerging areas of biology and biomaterial science. The first speaker is Tuo Wang. Tuo obtained his PhD and conducted his postdoctoral work first at Iowa State University and then at MIT. And he started his own lab at Louisiana State University in 2017. Since then has been doing pioneering work in applying DNP sensitivity enhanced solid state NMR to understand the glycan-rich cell walls of fungi and plants. Tuo, the floor is yours.
Okay, so let me share the screen. Okay, so can you see the presentation? Yeah. Okay, I would like to thank May for the nice introduction, and I would like to thank all the organizers for this valuable opportunity. So today we will share with you our progress using solid state NMR and DNP to elucidate the carbohydrate structure in plant biomass and fungal pathogens. And you can see the beautiful LSU campus, next to the Mississippi River here, and the chemistry and the material building. So before I start, I want to thank all the people who made the research possible, especially my current group members. And I want to thank the very strong instrumental support from National Mag Lab and EMSL. So I would like to thank Dr. Mei Hong for 7 years of training in biomolecular NMR and our current collaborators for a beautiful discussion. And finally, I want to thank NSF, NIH, and the DOE for funding support. So at LCU, I have been developing a comprehensive research program with a high focus on carbohydrate. For example, the biomedically relevant fungal pathogens, the bioenergy important plant biomass, and we also have methodology development, basically natural abundance DNP and the database. So I always receive the same question. So why complex carbohydrates? So what's special part with this molecule that makes you so excited? So there may be many answers. For example, okay, the systems are highly complex but similar in structure, as you can see from the representative carbohydrates in plants, in fungi, in algae, in bacteria, and in mammalian cells. And it's a very challenging system to study. And there's naturally a technical bottleneck because they are difficult to characterize in their cellular environment and also result in a knowledge gap. And at the same time, those molecules are involved in many biochemical and biomedical and bioenergy systems. For example, they're important for cellular recognition and they're important storage molecules. So they are intrinsically important. So besides those reasons, there's only one single reason that drives me towards this direction. So the carbohydrates are spectroscopically beautiful. So they give very beautiful NMR spectra. And I hope today I can convey the beauty of those molecules to you by two stories. So my first independent story is to reveal the fungal cell wall architecture in the purpose of promoting drug development. And the focus is this beautiful but deadly fungus called Aspergillus fumigatus. Fungal infection is very common. Superficial infections happen to more than 25% of human beings on Earth, for example, isolated foot and ringworm. Then you may be wondering, why does it matter? Yeah. So you may not be aware that life-threatening infections also occur to more than 2 million patients annually, with a high mortality of 20 to 95%, depending on the fungal species. So those infections can happen to the lungs, to the brain, to the kidney, or to the skin. And taking the Aspergillus as an example. So this is the airborne fungus, which means we breathe in the spores, every day. And for individuals with immune deficiency, so the fungi will localize to their lungs and form huge fungal balls, as revealed by the CT here, and finally lead to death. So recent efforts have been devoted to developing antifungal compounds that target the carbohydrate components in the cell wall. And that's because the polysaccharides in the cell wall, they're unique to the fungi. They are absent in our human cells. For example, we have chitin, which is a nitrogenated sugar that forms the microfibrils through the hydrogen bonding systems between the carbonyl and the amide groups. And we have beta glucans, which are the major building units that cross-link different polysaccharides. And they can be highly diverse in their covalent linkage. For example, we have 1,3, 1,4, so 1,4, and then 1,6 covalent linkage here. And we have the mysterious molecule called alpha-1,3-glucan, whose structural role was unknown. And it has a putative function for concealing, so for hiding the beta-glucans from the detection of the beta-glucan receptors on the host cell. So the fungal virulence will be increased. So how should we characterize those systems since they are highly complex and they are whole cells? Yeah, the strategy is first we carbon-13 and nitrogen-15 label the samples, by feeding them with isotopes. And the sensitivity will allow us to measure many 2-dimensional and 3-dimensional correlation spectra to study the structural polymorphism, intermolecular packing on the sub-nanometer length scale, site-specific hydration, molecular motion, and the domain distribution. And the information will be combined to give a view of the 3-dimensional architecture. This is a 2-dimensional carbon-carbon correlation spectrum measured on an 800 megahertz NMR on the carbon-13 nitrogen-15 labeled Aspergillus fumigatus. After preparation, the whole cell sample is directly packed into the rotor without any treatment. And the carbon-13 long width is 0.4 to 0.7 ppm for rigid components and 0.3 to 0.5 ppm for mobile components. So the resolution, I would say, is amazing, considering that we are handling whole cell samples of living fungi if you pack them freshly. With a great resolution, we're able to assign the signals from 7 types of polysaccharides and 23 conformers. For example, we have chitin here, the nitrogenated sugar, and we have alpha-1,3-glucan, beta-1,3, 1,6, and 1,4 glucans. And the color code and the short names will be kept for the later slides. I will especially guide your eyes to this well-resolved peak. So it's a chitin carbon-1 to carbon-2. And the chitin carbon-2 is covalent linked to the nitrogen in the amide group, which gave it a unique chemical shift at 55 ppm, just like a protein backbone. Yeah, and this peak and this chemical shift further correlating with 103.6 ppm of the carbon-1, giving this well-resolved chitin carbon-1 to carbon-2 cross peak. We were already highly satisfied with the data at that moment, but later, we got the chance to try those samples on the 1.5 gigahertz world record magnet at MagLab, and we are shocked by the further improvement in the resolution. If you compare the two spectrum, you can see that all the round circles on the 800 megahertz now become tiny dots. Yeah, you can see 3 dots here, you can see 2 dots here. The resolution, the carbon-13 long width is 0.3 to 0.5 ppm even for the rigid component. And with 10 milligram sample packed in a 2 millimeter rotor, we're able to finish a 2D carbon-carbon and a nitrogen-carbon correlation spectrum within 2 hours each. And this picture shows the beautiful magnet at 35 Tesla. So polysaccharides, they are very different from proteins. So the fundamental units, basically the monosaccharides, they are highly similar in structure. So in spectroscopy, that will result in many highly crowded clusters of cross peaks. And you can directly visualize it from the spectrum. And with the great resolution at 1.5 gigahertz, we can resolve tons of cross peaks within each small box in the spectrum, not only for the microcrystalline chitin, but also for the disordered glucans. So the key question is how those molecules pack in space to form the cell wall. And the overlay of the long-range PAR spectrum in black with the short-range DAR spectrum in magenta allows us to resolve tons of intermolecular cross peaks. And most of those cross peaks are between 2 molecules, alpha-1,3-glucan and chitin. For example, between alpha-1,3-glucan carbon-1 to chitin carbon-4 and to chitin carbon-6. So here is the alpha-1,3-glucan carbon-1, and that's the carbon-4 and the carbon-6 of chitin. So the data indicates that the chitin and alpha-1,3-glucan, they tightly pack to form the structural core of the fungal cell wall. So DNP enhances NMR sensitivity by tens to hundreds of fold by transferring the polarization from electrons in the radical to the NMR active nuclei in the biomolecules. And we successfully obtained 30-fold sensitivity enhancement on the whole cell fungal sample. And the DNP spectrum for the whole cell samples are typically highly crowded. Yeah, this is partially due to the large number of biomolecules in the sample and partially due to the coexistence of short range and long range correlation cross peaks. And by marrying 2D nitrogen-carbon correlation spectrum with a short followed by a short carbon-carbon mixing, we only probe the intramolecular correlations. And with the long mixing, we simultaneously probe the short range and the long range correlation. And the spectral subtraction will give us a different spectrum that only report the intermolecular cross peaks, which are structurally meaningful. For example, between chitin nitrogen to alpha-1,3 glucan, carbon 1, and carbon 2. With only 3 milligrams of the fungal material and with only 0.1 to 0.2 milligrams of the chitin, we're able to finish those experiments within 13 hours and obtain many intermolecular cross peaks. So by combining the information on the intermolecular packing and the data on hydration and the dynamics, which are not shown here due to the time limit, we're able to construct a new structural model of fungal cell wall. So alpha-1,3 glucan and chitin, they tightly pack to form hydrophobic and rigid cores that are embedded in a well-hydrated and mobile matrix formed by diversely linked beta-glucans and further capped by an outermost very mobile layer rich in glycoproteins. And the structural wheel here is a combination of the molecular evidence that we obtained here using NMR with many biochemical studies in the existing literature. So the structural model serves as the basis for us to further investigate the biomedical questions related to structural diversity, fungal virulence, and drug effect. For example, you can see that the different fungal pathogens have distinct NMR fingerprint for the cell wall. And our preliminary data of the drug effect is that, okay, by adding the antifungal compound caspofungin, all the signals, most of the signals from beta-glucan is gone. Yeah, and also we have substantially reduced the hydration level for the remaining molecules as revealed by the decline in the water additive intensity. So the big picture here is by the treatment of this antifungal drug, the fungi start to stop biosynthesis of the beta-glucans, which are mobile and hydrated. And then the remaining part is only hydrophobic, basically chitin and alpha-1,3 glucan. So in this way, the water-soluble antifungal drugs will be inaccessible to the fungi, and the fungi can survive through the drug treatment. So now I will change the flavor to application with importance to bioenergy and biomaterials, and we will study the interactions between polysaccharides and lignin in plant biomass. And the pictures here show you the samples that we use. They are the stems of the maize, the corn, Arabidopsis, and the switchgrass. So plant cell wall is an energy-rich biomaterial that provides support and rigidity to the cell and regulates the plant growth. And this stores energy as carbohydrates. And in the primary cell wall, the fast-growing part of the plant, we have cellulose microfibrils embedded in the matrix of polysaccharides called pectin and hemicellulose. In the secondary cell wall, the material part, and the majority of plant biomass, we have the additional deposition of aromatic polymer called lignin. The interactions between the aromatics and the carbohydrates will make the cell wall resistant to enzymatic and chemical treatment. So they lock the energy in the system. So our goal here is to determine the chemical and the physical principles for the interactions between the aromatics and the carbohydrates. So using carbon-13 labeled maize stem, so the corn, the stem of the corn, and we are able to measure 2D carbon-carbon high-resolution inadequate spectrum, and you can see many types of cellulose signals, in red color. And cellulose is known to have surface glucan chains and internal glucan chains, who differ by the conformation, by the local conformation as well as the hydrogen bonding system. And the multiple cellulose microfibrils could possibly aggregate to form larger bundles. So you will have many different environments for the glucose units in the microfibril. For example, you have the deeply embedded core, you have an intermediate layer sandwiched between the surface and the core, and you have a hydrophobic and hydrophilic surface. So those structure features are resolved by the signals in the NMR spectrum as annotated using superscript from A to G. For the hemicellulose, we have Xylan, and then we have twofold Xylan signal in purple here, and we have threefold Xylan in blue, here. They are well resolved. So twofold here means you have every 2 sugar units for 360-degree helical rotation, and threefold Xylan means you have every 3 sugar units for 360-degree helical rotation. And you can see that on a larger length scale, you will have flat ribbon structure for the twofold conformer, but zigzag structure for the threefold conformer. So the lignin is chemically complex, and we can resolve all the beautiful aromatic signals from the fundamental units, like H, G, and S units, which have an increasing number of the methyl ether substitution. So OME here, you have 0, 1, and 2 OME in the structure. At the same time, we can use the carbon-carbon alpha-DR spectrum to resolve the covalent linkage patterns between the aromatics. So by the overlay of the short-range correlation spectrum in orange with the long-range correlation spectrum in dark gray gave us 74 intermolecular cross peaks in the maize sample, and in total 255 intermolecular contacts in 4 different plant species. And those interactions are between Xylan acetyl, to the cellulose, between lignin methyl to cellulose, between lignin and Xylan, and between different aromatic units in lignin, between lignin methyl to Xylan acetyl, and between Xylan acetyl and lignin. And it's noteworthy that the Xylan has a strong cross peak with the carbon-3 and carbon-5 of the S unit, so here. And the medium cross peak with the carbon-3 and carbon-4 of the G unit, but no corresponding cross peak with the H. So the interactions clearly correlate with the number of methyl ether substitution, and it indicates that the electrostatic contact between the polar motifs are crucial for stabilizing lignin carbohydrate interactions. And we have obtained a 23-fold sensitivity enhancement on the maize stem, with which we can integrate dipolar gating with frequency filter and microwave gating to cleanly select the aromatic signals against the dominant carbohydrate signals, which are 260-fold stronger. So I would like to mention that the DNP system at MagLab is equipped with a mechanical shutter, so it can turn on and turn off the microwave during each single scan for the clean selection. With the selected polarization from the aromatics, we further transfer it to the carbohydrate neighbors to detect the interaction interface. And the lignin bound polysaccharides clearly have different composition from the equilibrium state. You don't have signal from I. Yeah, which means you don't have signal from the internal chains embedded in the microfibril. And the surface chains with the cellulose also get decreased a lot. At the same time, it's interesting that the type A of the twofold Xylan, so this cross peak, doesn't show up in the Lignin-Edited spectrum. So it indicates that the flat ribbon structure doesn't bind lignin. So this finding echoes with an earlier study by Dr. Dupree's group, who has found that the flat ribbon structure preferentially binds the even surface of cellulose instead of lignin. So the dominant signal comes from the threefold Xylan, as well as the 2 twisted conformers of the twofold Xylan, indicating that the non-flat structure of the Xylan is binding the intrinsically disordered aromatic domains of lignin. So the Xylan-lignin interaction is conformation dependent. So NMR is a powerful tool for investigating molecular motions with all the well-resolved, well-established techniques. And here we mainly study the nanosecond time scale motion using carbon T1 relaxation measurement, and the microsecond time scale motion by measuring proton T1 row relaxation. And the lignin clearly has the longest carbon T1 relaxation times, but the shortest proton T1 row relaxation times. So this is understandable because the rigid structure of the aromatics, they are resistant to local rapid ring orientation happening at the nanosecond time scale, but they can have the ring flipping motion, just like those happening in the protein, tyrosine, and phenylalanine side chains, or they could have collective motions of multiple aromatic rings. So cellulose, in contrast, has the longest proton T1 row relaxation times. So it's because the systematic hydrogen bonding in the microfibrils prevents multiple sugar rings from having collective motion on the slow time scale. So this dynamical heterogeneity further correlate with the hydration profiles of our molecules probed using water-edited spectroscopy. So the method basically detects the well-hydrated molecules. For example, it detects the threefold Xylan signal in blue over the twofold Xylan signal in purple. And the quantification of the water-edited intensity will provide the site-specific hydration map. And the hydration level decreases from the threefold Xylan, the best hydrating molecule, to the twofold Xylan and the surface cellulose, two molecules with a tight association and similar hydration level, and then to the internal chains with cellulose and then to lignin. So we clearly have an issue here. Previously, we have shown tens of intermolecular cross peaks between lignin and the threefold Xylan. How could they still retain fully different dynamics and hydration? That can be explained by one type of structural model in which the aromatics with the lignin self-aggregate to form nanodomains that are large enough to retain their unique dynamics and hydration. And the nanodomains have extensive surface and electrostatic contact with the threefold twisted conformer of Xylan, which are further connected to the twofold flat ribbon conformer of the Xylan. And the twofold Xylan is further coating the surface with cellulose microfibril. So in this way, the hemicellulose Xylan is smart enough to bridge the two hydrophobic cores of the cell wall, cellulose, and lignin together. So while the previous studies are focused on the isotope labeled samples, and we also tried the natural abundance DNP on unlabeled systems, and we found the polysaccharides carbohydrates, they are perfect system for this technique. With a short measurement of 4.5 hours, we are able to measure beautiful spectrum on the unlabeled cotton sample, and the resolution is 0.9 ppm, and the sensitivity is just decent. And we can resolve all the glucose units, A prime, B prime, in 1 alpha and 1 beta cellulose. And with a total measurement time of 54 hours, we can probe the Xylan conformations in 2 unlabeled rice stems, as shown here, wild type and mutant. And we can see that the mutant has enhanced the conformation of the threefold Xylan, so that it means that the mutant promotes the interactions between Xylan and lignin, but reduces the Xylan cellulose contact. So all the information are obtained with a short measurement time of multiple days, and our friend, Dr. Fred Montingo-Wegener, is helping us with those experiments. So at the same time, to accommodate the growing data set on carbohydrate, we have coded the Complex Carbohydrate Magnetic Resonance Database, www.ccmrd.organization. So the database is currently under upgrade and maintenance, and will be back to service very soon with new functions. So I will stop here. I want to thank all my group members, and I want to thank you for your attention, and I'm ready to take questions. Yeah, so I cannot hear you.
Yeah, thank you, Tuo,
thank you.
for interesting talk. So I see 2 questions coming up, and there's also a chat. The attendees, please type your questions into the Q&A part. So the first question is, how do you label fungi cell wall?
Yeah, so our first samples are purchased directly from a labeling company, but all the later samples, we prepare them freshly in lab. So it's very easy. You just feed them with the carbon-13 glucose and the nitrogen salt. Depending on different fungal species, they have different salt to use. Yeah.
Great. And the second question is by Hashim. For first part of the talk, how much would the NMR spectra and the correlations differ if you were to isolate the carbohydrate component from cells and look at them in vitro? Does the cellular context make a difference?
Yeah, actually, that's something that we are currently doing in collaboration with Dr. Jean Paul at France. So, and then the key conclusion is, okay, the major chemical shift doesn't change. Yeah, they have some minor variation, but it's within like a 0.2, 0.3 ppm range. Yeah.
Okay. All right. Next question is by Alon's Lens. Did you try to record inept-based experiments to probe more dynamic part of fungus?
Yes, we have done this, but most of the signals come from the intracellular components. So small sugars, lipids, and something else. So we care more about the structural component of the cell wall part. They are rigid. Yeah.
All right. Next question, anonymous. Is microwave gating necessary? How would leaving the microwave on all the time affect the selectivity?
Yeah, so if we leave the microwave on, then we had a really hard time to get the clean selection because the carbohydrate is 260-fold higher than the aromatics. So any leftover of the magnetization will dominate the spectrum. Yeah, so without the microwave, then we will avoid the repolarization with the carbohydrates. Yeah.
All right, good. Next question is Tanguy Lebron. Thank you for your very interesting presentation. Can specific labeling of carbohydrates reduce the complexity of 2D carbon-13 spectra?
Yeah, we just purchase as isotopes. Yeah, good question. Yeah.
All right, next question is by Rosalie Thompson. Do you find significant broadening with DNP with the lower temperature? And, oh, this is flashing on me. Do the radicals or the process to add the radicals disrupt the structure at all?
Yeah, so these actually two questions. So first, so when we at the DNP condition, the rigid components, for example, chitin, fungi, and cellulose in the plant, they doesn't get broadened a lot. So they keep the beautiful resolution. The mobile components, they get broadened. Then the second question is, okay, adding the radical, we have compared the spectra before and after adding the radical, this keep the same envelope. Yeah, so but the mobile components for sure get broadened. Yeah.
Mm-hmm. Okay, next question is by Atul. The name is too long for me to read. Okay, the other biomolecular components of the cells seen in the NMR spectra, that's one. Do they appear at different positions relative to the carbohydrates? And is that why the carbohydrates can be visualized without crowding from other biomolecules?
Yes, we actually have a figure that I didn't show, so we have all the proteins, we have the lipids, and we have the carbohydrates. They have their own signature. Yeah, they have their own chemical shift, well-resolved regions.
Okay. All right, as another question by Gigi, did you have to initially calibrate your chemical shifts to the solution carbohydrate chemical shifts?
So we didn't calibrate to solution carbohydrate chemical shift. We just use the TMS standard. And then it's interesting that if you have very mobile components, they have similar chemical shift as the solution state. If you have rigid components, usually they don't overlay. Yeah. Yeah.
Okay. So that's an internal calibration. All right. Okay. So here's another question. If you're using glucose to label the cell wall, will you get signals from proteins in membrane?
Yeah, we get all the signals. Yeah, they're well-resolved regions. Yeah.
So let me follow that up. Follow up with that question. Then you just didn't show us the protein region, or in
we actually show them in the paper, so we didn't show them here. Yeah.
Mm-hmm. Okay. So here's another question, Valeria. Have you noticed any peak that appears only in solid state and not in solution state?
Actually, all the signals, if you talk about the rigid components, they also show up in solution state, but usually they are like already destroyed partially. Yeah, so they are like digested and then solubilized. So their signals doesn't overlap that well. Yeah.
Okay. Here's another question. Very nice talk. Can you use solid state NMR to resolve branch structures in glycan?
So branch structure in glycan, we can identify at which position the glycans are branched. So it will induce a unique chemical shift change. So we have done this previously, not with the glucans, but with the arabinine. Yeah, so we can see all the branching details. Yeah.
So let me ask a question. So have you compared the relative merits of 2D inadequate spectroscopy versus potentially 3D carbon correlation in terms of the resolving power and information content of these material?
So we didn't, actually we haven't measured 3D spectrum on the fungi. We recently measured some 3D on the plant. So the key point here is if we can resolve them very well, we don't need a 3D. But for the 3D, we use them more for the structure determination, yeah, to get long-range correlations, yeah.
Okay, I think we had a good set of questions. I hope I'm not missing anybody else's. Oh, here's another question. Can fungus still grow after spinning it?
Exactly, so that's a good point. If you measure them freshly, take them out of the rotor, they should be able to grow. Yeah, so they're alive. Yeah.
Okay. All right, thank you, Tuo.